some time now. And this really sort of stems from the idea that it was proposed back in the late 1970s by Melzack and Losa. And they basically suggest, after they looked at a whole bunch of individuals with spinal cord injury pain, and they found that you could perform a whole bunch of surgical and non-surgical interventions, such as rhizotomies, sympathectomies, and they actually went in and did chordotomies, and they found that that didn't relieve the chronic pain in about a dozen individuals with below-level spinal cord injury pain. So they proposed that maybe the periphery, at least in some neuropathic pain conditions, has very little to do with the ongoing or the maintenance of chronic pain. And they basically suggested that there was a pattern-generating mechanism within the central nervous system itself. They suggested it was above the level of the dorsal horn. I don't know whether it is or not, but they certainly suggested that it could be maintained with central nervous system activity itself and have influences from a whole range of factors, including cultural factors, gender, visceral inputs, stress, all sorts of things. But they basically were suggesting that it's this pattern-generating mechanism that's important that we should try and understand. So we've been looking at this for about 6 or 7 years now or more, and we've been thinking about where would you actually look for a pain generator if there was one. So of course this is a diagram that you're familiar with, where we know noxious information comes in, synapses in the dorsal horn, it then contacts second-order neurons which travel across the midline and up to the thalamus and up to the cortex. So this spinothalamic pathway is very well documented. It's not obviously the only pathway that transmits noxious information, but it's probably the one that's important in the constant perception of a noxious stimulus, or the pain itself. So if you were going to sort of follow that road, there's a couple of curious things which occur in some individuals. And the interesting thing about central nervous system lesions, or natural lesions, or strokes, is that if they occur in the somatosensory part, in the spinothalamic pathway, you have a good chance of developing post-stroke neuropathic pain. And here are two individuals where there's a tiny little stroke in the thalamus, there and there, and this isn't the only two that they looked at. They looked at a whole bunch. And this seems to be localised in the somatosensory thalamus, so where the spinothalamic tract actually, or where part of it terminates. And also, you can get neuropathic pain following a lateral medullary stroke. So here are 3 individuals with a lateral medullary stroke, and that's from a blockage of the vertebral artery, or PICA. That's not an uncommon condition. And if it actually includes the spinothalamic tract, there's a good chance that they also can develop neuropathic pain. And there's also some evidence after spinal cord injury that if you damage the spinothalamic tract with the spinal cord injury itself, you're more likely to develop neuropathic pain after spinal cord injury. So there's good evidence if you lesion or if you have a stroke within the ascending spinothalamic tract, you can develop neuropathic pain. It very rarely happens with a cortical stroke. The insula sometimes results in a chronic pain condition, but it's not that common that cortical stroke can result in neuropathic pain. It's far more common if you damage the spinothalamic tract. So I guess we started to look at the spinothalamic tract in individuals that don't have a central neuropathic pain condition, but more of a peripheral neuropathic pain condition. And I'm just going to present some of the stuff we've been doing on orofacial pain today. And of course, if you look at the orofacial system, it doesn't come in at the dorsal horn, it comes in at the spinal trigeminal nucleus. And it's a little bit of a more complicated pathway, but it basically crosses the midline again up to somatosensory thalamus and up to the cortex. So what we were interested in firstly some time ago was to see if we could see if there were anatomical changes in that pathway in individuals with neuropathic pain. Might be the UFO landing, I don't know. So what we did was we started to look at individuals with orofacial neuropathic pain, and these individuals all have pain following endodontic work. The dentist never tells you that when you go and you have a root canal, there's about a 10% chance you'll get chronic neuropathic pain. They sort of fail to tell you that, which is rather unfortunate. But there is a good chance you'll develop neuropathic pain after a root canal. And all of these individuals actually had that post-endodontic work neuropathic pain, and they're all quite long-term. They all had it for at least a couple of years. Some of them had it for about 10 or 12 years, and it's often quite severe. So I guess the nice thing about looking at the orofacial system is you can look at the primary synapse. So you can look at the spinal trigeminal nucleus in humans. In humans, you basically cannot look at the dorsal horn using brain imaging. It's extremely difficult and you just can't do it. It's too small. But you can certainly look at the spinal trigeminal nucleus using brain imaging. At least now you can. So there are a number of ways in which you can detect anatomical changes in living individuals using MRI. The first one is called voxel-based morphometry, which is probably the oldest technique. It's been around for 15 years or so. And what this technique does is it's very simple. You basically get an individual's anatomical image. You collect one of these every time you put someone in a scanner. It's called a T1 weighted anatomical. You warp it to a template brain made out of 300 Canadians, and you basically then look at how much warping. So it's supposed to be relatively normal, this template. And so if the area that you, if an area needs to shrink, if I need to shrink the area to match the template, it was larger to start with. And if I need to expand the bit of cortex to match the template, it was smaller to start with. So it's very crude, but it's a really, really good way to sort of start to have a look. And we did that in individuals with our neuropathic pain, orofacial neuropathic pain, and we found primarily decreases in brain volume, or grey matter concentration is what they talk about. And we found it in nucleus accumbens, we found a bit in putamen, but the main thing we found was, the most significant change was the change in the thalamus. So it was a decrease in volume in the thalamus. And of course that got us really excited, because if you lesion that area there, you can develop neuropathic pain. So we were thinking, well, maybe after a peripheral nerve injury, you get changes that work their way up the chain from the primary synapse up to the thalamus, and then you end up with a chronic pain condition due to those anatomical changes. I'll talk about the spinal trigeminal nucleus in a second. And one of the ways in which you can try and identify whereabouts in the thalamus that is by simply brushing the orofacial region. So that should activate the somatosensory thalamus, the same area that gets noxious input. That's a very simple idea. You get activation. So we know some of this anatomical reduction actually encompasses the somatosensory thalamus. There's some in other areas. There's probably a bit in centrolateral, medial dorsal, but there's certainly some overlap into somatosensory thalamus. And that's nicely correlated with the amount of pain they have. So the greater the grey matter loss there, the more pain they had. It wasn't correlated to duration. Okay, so that was sort of our nice first step that we did quite a while ago. Now, the biggest problem with voxel-based morphometry is no one has one clue what it means. They all tell you they know what it means, but no one has a clue. It just means there's a change in volume. In fact, it can mean there's a change in sort of concentration or density. So it's a really good way to start, but it's a very bad way to finish. So, but the beauty of doing imaging is that within the scanner itself, you can do a whole bunch of stuff. You can look at water movement, you can do spectroscopy, you can look at blood flow, you can look at changes in bold imaging. So there's a whole range of things that you can sort of add to the mix to try and work out what that decrease in volume actually represents. So what we did was we started to use a technique called magnetic resonance spectroscopy, which uses the same machine, just a different button. And what that technique can do is it can actually give you a measure of the biochemistry within an area of interest. And there's a number of different MRS scans that you can do, but the most standard one is this sort of proton magnetic resonance spectroscopy. And that's about the smallest box that you can measure from using a standard 3 Tesla scanner. Now that they've got 9 Tesla scanners, you could probably measure something about probably a 20th of that size. But at the time, that was sort of the minimum size. So you can see it's quite big. And the limiting factor with this MR spectroscopy is that you really do need to know where you want to look. With voxel-based morphometry, you just scan the whole brain and it tells you what's different. With MRS, you need to say, okay, I'm interested in area X or area Y. Anyway, so we basically did that, and you get this nice squiggly line, and whereabouts it is on this parts per million axis will give you the actual chemical itself. And basically, the height of the curve gives you the amount of the chemical. So we did that in our orofacial pain patients and we compared it to our control group. And one measure that biochemists came up with is a measure of NAA over creatine, and that is apparently a measure of neural viability or health. So if you get a reduction in this NAA over creatine ratio when you actually measure those chemicals, it can indicate that there's a change in neural function, and the health of the neurons are lower, or they're worse off. And the nice thing about it was that on the side ipsilateral to their ongoing pain, so the side that doesn't receive the noxious inputs of the spinothalamic tract, there was no, sorry, I should have said trigeminothalamic tract, there was no change in neural viability, but on the side opposite to the ongoing pain, there was a significant reduction in that ratio. So this is some sort of type of evidence that that change in brain volume or grey matter volume actually means something, and we actually have a change in some type of neural function. So that sort of still goes into the idea that maybe anatomical changes at the level of the thalamus at least could be involved in maintaining neuropathic pain. Okay, so that's quite nice, and it sort of marries up with the old stroke data, although these are tiny and this here is quite large, I give you that. But they sort of seem to be matching up quite well. So, I mean, that's interesting, but it doesn't mean anything unless it does something to the brain and changes your brain activity. So if it doesn't change brain activity, it means nothing. So what we did was we thought, well, okay, is this associated with a change in activation or a change in ongoing flow within pain areas? That's, I guess, my naive assumption. I was completely wrong, but I'm often wrong. I was just really wrong on this account. And one technique you can use to measure that is called arterial spin labelling. And this is actually quite a new MRI technique where you can actually measure basal blood flow. Until sort of 4 or 5 years ago, you could only measure a person's ongoing blood flow by using a PET scanner, basically. And so it is really difficult to compare a population of patients with any type of disease with a control group using fMRI. You can't. fMRI gives you a relative measure. So you actually need to give somebody a stimulus. So if I was interested in chronic pain and controls and in comparing their brain activations, I couldn't look at it at a static level. I couldn't just say this area was increased or decreased relative to controls. I'd actually have to do something to them, which is extremely limiting. But this technique gives you a nice measure of blood flow. So ongoing blood flow, which is coupled to neural activity. I don't know why this hasn't taken off, but anyway, it hasn't. So you can put someone in a scanner and 6 minutes later you have a nice blood flow map. Now the interesting thing is, if you were like me and being very naive, you would assume that, okay, well if I've got chronic pain, I've probably got increased activity in thalamus, S1, S2, cingulate, all those areas that people tell you are involved in acute pain processing. And that's exactly what you find if you do an acute pain study. So you get increases in blood flow in those areas, so that's quite nice. The problem is you do not get that in neuropathic pain patients. In fact, you get nothing like it. What we found was that we found really only about 2 or 3 areas where you get a change in blood flow at all in these patients. And one of the areas was actually a decrease in blood flow in the thalamus, not in the somatosensory thalamus. We found a decrease in blood flow in primary somatosensory cortex, and that's about it. And if you look at the literature, that's actually quite consistent. I should have looked at the literature before I started, but I didn't. There's about 4 or 5 studies out there that actually show that if you put somebody in the scanner with neuropathic pain and you measure ongoing blood flow, you basically don't get increases in activity or increases in blood flow in acute pain circuitry. The most consistent report is actually a decrease in thalamic blood flow. And the interesting thing about it was that it wasn't actually in somatosensory thalamus, it was right on the edge of the thalamus. And this area of the thalamus is a long strip of neurons, and they form what is called the thalamic reticular nucleus, and seem to overlap that, which is interesting because that area of the thalamus actually receives input from somatosensory thalamus, and it regulates all sensory information going through the thalamus to the cortex. And this is just from a monkey, and this is the area of the thalamic reticular nucleus that basically gets somatosensory input. It's very similar to the area that we found a decrease in blood flow in humans. So that was interesting, as well as the fact that you get a change in primary somatosensory cortex in the area that receives information from the ongoing pain area. So I thought you'd get an increase in blood flow in thalamus and an increase in blood flow in S1, and we didn't find that at all. And we double-checked, triple-checked and quadruple-checked, and it's still coming out with exactly the same thing. And I mean, that was a little bit disturbing, but it's actually not that disturbing, because if you go and look at the literature, when you're looking and you start to look at the thalamic reticular nucleus and its role is to regulate all sensory information, basically going from the primary sensory nuclei in the thalamus up to the cortex. And it's involved in, sleeping and general arousability. So it has very distinct sectors there are basically no overlapping sectors. So somatosensory part of the thalamic reticular nucleus will be next to the auditory sector, will be next to the visual sector, and they basically do not overlap. And this circuitry is probably one of the most well-described circuitries in the whole human brain. If you actually start looking at the literature, there's thousands of studies looking at this circuitry. And what this circuitry involves is an afferent that goes up from the thalamus, say the somatosensory thalamus, up to the cortex. It gives off a collateral to the thalamic reticular nucleus, and at the same time, the same area of cortex projects back to the somatosensory thalamus, and it also gives off a collateral to the thalamic reticular nucleus, and that projects back to the somatosensory thalamus. And that neuron that projects back to the somatosensory thalamus is GABAergic. And all of the nuclei within the thalamic reticular nucleus are GABAergic. There's not a single neuron that's not GABAergic. So you've got this shell of neurons that surround sort of the outside of the thalamus that are inhibitory, that project down to the thalamus and actually regulate everything that's going on. And they're involved in setting brain rhythms. And basically, there was an old idea by Crick who suggested that this was like a searchlight hypothesis. So it was like a searchlight in the dark. So the thalamic reticular nucleus is a big searchlight searching through the dark sky, and it's basically funneling information and making you focus on particular types of information that are relevant and dampening down information that's not. And it's sort of funneling that information to the cortex. Now, there's a few studies which show that in neuropathic pain patients, they have an alteration in this thalamocortical rhythm. So the way in which the thalamus is talking to the cortex is significantly altered. And they've found that there's an increase in sort of this thalamocortical power. So there's an increase in the way in which neurons oscillate and the way in which they talk to the cortex in sort of a broad sense. And that fits extremely well with this sort of model of the thalamic reticular nucleus, where you've got a dysregulation of this circuitry, and that results in thalamocortical dysrhythmia. Whether that thalamocortical dysrhythmia gives you the constant perception of pain is another story, but you certainly get thalamocortical dysrhythmia in these individuals. I don't know why. No one really knows why. They've speculated, but no one really knows why, but it seems to be a common property. Anyway, so given this change in thalamic reticular nucleus blood flow, we were thinking, well, how can we actually measure thalamo-reticular nucleus output? And one of the nice things about the thalamic reticular nucleus is it's jam-packed full of GABAergic neurons. There are other GABAergic neurons in the thalamus, but by far the largest amount are in the thalamic reticular nucleus. So you can go back to your old friend and use spectroscopy and try and measure the amount of GABA in the thalamus in these chronic pain patients. Now, GABA spectroscopy is an absolute nightmare. There's no other way to say it. It takes about half an hour to collect a single voxel, and that's the size of the voxel. It's about 2 centimetres by 2 centimetres by 2 centimetres, and it takes half an hour. It's a very low concentrations, and it's very difficult to detect. As magnetic field strengths get higher and higher, that sort of plot gets wider and wider, and the GABA peak will become larger and larger, so it becomes a lot easier. But at 3T it's very difficult. But the nice thing about it is that most of the GABA in here is actually coming from the thalamic reticular nucleus, so I don't really care if I've got other parts of the thalamus. I'm pretty sure if I get a change in GABA, it's coming from the TRN. And that's exactly what you get. You get a massive reduction in thalamic GABA in those orofacial pain patients. And we've done exactly the same in spinal cord injury with Paul Wrigley, and you get exactly the same change in blood flow in spinal cord injury and neuropathic pain, exactly the same change in GABA levels within the thalamus. So we were sort of working our way into a bit of a model, but I guess the next step that we're interested in, you don't get an increase in blood flow in S1 or thalamus or anything like that, but if you actually look at the amount of GABA in the thalamus and you look at how the thalamus talks to the cortex, so you do use a technique called functional connectivity, which is slightly dangerous if you don't know what you're doing, and you should read every functional connectivity paper with a little bit of a grain of salt. But what it does, it's simply trying to measure covariation between signals between two areas. That's all it's doing. You'll get a signal fluctuation in one area and you're measuring a signal fluctuation in another area. How tightly are they coupled? It doesn't tell you one area goes to the other area. It could go via 14,000 other areas before it gets there. But based on the neuroanatomy that we know, you can make, judgments and hypotheses based on that. And the interesting thing about that is if you then look at the amount of GABA within the thalamus, it's directly related to its connectivity with the cortex and areas of the cortex that are involved in acute pain processing. So you get a change in connectivity with primary and secondary somatosensory cortices, and also you get some in the insula. So it looks like the amount of GABA within the thalamus is actually influencing the way in which the thalamus talks to the cortex. Now we know that with thalamocortical dysrhythmia. We know when you measure an EEG you get thalamocortical dysrhythmia, so the thalamus is talking to the cortex differently. But this sort of low frequency fMRI study shows you that even at low frequency it seems to be related to the amount of GABA within the thalamus, or the amount of GABA loss within the thalamus. So we decided to sort of come up with a very naive model where you basically get a nerve injury that somehow works its way up to the thalamus. You get a reduction in the way in which the thalamus talks to the thalamic reticular nucleus, so you get a decrease in blood flow there, you get a decrease in blood flow in the cortex. That basically affects this circuitry. It reduces the amount of GABA in the thalamus. You get orthothalamocortical connectivity, and I don't know how, but somehow that results in a constant perception pain. Yep.
When you say a reduction of GABA, do you mean outside cells or inside cells, or can you tell?
You can't tell.
So it's just flooded in and out?
Whatever GABA is in that voxel is what you're measuring, wherever it is. In a half an hour, in 2 centimetres by 2 centimetres by 2 centimetres.
Okay, down as well.
Yep.
Right.
Yep. I get it. I realise that. Yeah, I mean, that's one of the things that is difficult. Okay, so the other thing that's quite interesting is if you look at the literature and you look at the thalamus in humans with deafferentated neuropathic pain, there is some old evidence that shows that you get a change in bursting activity within the thalamus. So you get a change in thalamic firing. Instead of it being relatively constant, I should say, you basically get really quick bursts of activity in individuals with neuropathic pain when they're awake, and you don't get that in controls, or you get less of that in controls. So there was a nice animal study which showed this really nicely after a chronic constriction injury, and this is a study where they basically measured ongoing neural activity in the ventroposterior thalamus, the somatosensory thalamus. They performed a chronic constriction injury in the sciatic nerve, which is the model of neuropathic pain, and they looked at ongoing neural activity, and they found this very low frequency oscillatory pattern within the somatosensory thalamus. And it doesn't look like that when you measure it in a control rat. So not only do you get sort of these anatomical changes, but you're also getting a change in the way in which the neurons fire. And these low frequencies oscillations are very low frequency. They're about ongoing up and down about every sort of 50 to 100 seconds. So they're very infraslow oscillations is what they are. So we were interested in this because there is an idea about thalamocortical dysrhythmia that it's basically a mismatched interaction between very low frequency or low frequency oscillations and high frequency oscillations. And that's how you get this sort of thalamocortical dysrhythmia. It's basically the ongoing, when they talk about low frequency, they're talking at 5 Hz. there's an interaction between sort of 5 Hz and higher Hz frequency activity that doesn't occur in individuals that don't have neuropathic pain. And that's what they think causes thalamocortical dysrhythmia. But there's also an increase in really low frequency oscillations in neuropathic pain patients. In animal models of neuropathic pain. So we were interested in seeing whether that was also the case in the thalamus in our individuals with orofacial neuropathic pain. So one way in which you can do that using functional MRI is to measure resting state fMRI. So you put them in the scanner, you get them to lie there for a while, and you basically just turn the scanner on. And then you can measure low frequency oscillations in blood oxygen level dependent fMRI, so low frequency oscillations in sort of blood flow. And then what you can do is you can get those signals out and you can basically perform a fast Fourier transform, which is a very simple procedure just to break it down into its individual components. And then you can plot a frequency power spectra and you can see if there's an increase in low frequency oscillations in your humans with chronic neuropathic pain. Now, there's some really nice software to do this, but I didn't want to make it easy for an honest student. So what I got it to do was, one of the issues is you don't really know which frequency band to look at. And if you look at the entire frequency band, you could be missing something. In fact, when you look at the entire frequency band that you collect, which is up to sort of 1/4 of a hertz with functional imaging, you basically get nothing. But if you start to sort of put a grid through the thalamus and then look at every single voxel, I got it to look at 240 and do fast Fourier transforms on 16 patients and 40-something controls, then there's a really nice little sort of hump of significant differences around sort of 0.03 to 0.06 hertz. So you can then use this nice software which will basically look at that low frequency in every single voxel throughout the brain and tell you for every single voxel, is there an increase in power at that low frequency in your patients versus your controls. And if you do that, you surprisingly get an increase in low frequency power in all of the usual suspects. So you get an increase in low frequency power in the thalamus. I've said this is somatosensory. It certainly overlaps with somatosensory, but it also includes centrolateral. You get an increase in these low frequency oscillations, just like they found in the animal experiment. You get an increase in low frequency oscillations in the thalamic reticular nucleus in exactly the same spot where you get a decrease in blood flow. You get an increase in primary somatosensory cortex, and really nicely you get an increase where those afferents come in at the spinal trigeminal nucleus. So you get a massive increase in these low frequency oscillations at the level of the orofacial equivalent of the dorsal horn, which is really, really lovely. And the other thing is you don't get that if you give someone acute pain stimulus. So if you inject hypertonic saline into the masseter muscle, for example, and give a constant noxious input, and you do exactly the same thing, you don't get any increase in infraslow oscillations in those areas. So those increases in low frequency fluctuations have got nothing, are unlikely to have anything to do with afferent drive, we think. Okay, so one of the interesting things about those low frequency oscillations is there's been a lot of work on astrocytes looking at low frequency oscillations at that sort of level. And I put this up because I always like to put up the neural threesome. I thought I'd have any chance you get to use that word, you've got it. So I thought I'd do that. So we all know that astrocytes basically engulf the synapse, so they can regulate neurotransmitter uptake and all that kind of stuff. But of course, over the last 20 years, people have realised that they also release neurotransmitters and modulate the synapse. And they release gliotransmitters, they release glutamate, they release ATP, they release GABA, and people think that they actually modulate the sensitivity of that circuit of all synapses. And the other interesting thing is that they're connected, probably via gap junctions, maybe, but they certainly do oscillate together, or they certainly can oscillate together. And they have really nice calcium waves, and these are in vitro, so they're not in vivo. But they have a really nice spread of calcium wave, which will go from one astrocyte to the other. And there is an idea that Well, if you measure these astrocyte calcium waves, they can sort of form this irregular pattern, or they can be quite regular in nature. And there is an idea that in some pathological conditions you get astrocyte activation, you get an increase in these calcium waves, and that they can basically start to oscillate together. And they can form sort of one big oscillating mass. And that big oscillating mass can then regulate receptor function and regulate the synapse itself. And they basically are regulating at exactly the same frequency that we find changes in low frequency oscillations in those pain pathways in our neuropathic pain patients. So there is really good evidence actually that astrocytes are activated in animal models of neuropathic pain. There's hundreds and hundreds of studies that show that when you do a CCI, or if you do an infraorbital nerve lesion, you get a massive astrocyte activation where the afferents come in and terminate. And this is a nice study which just shows that. And they get really big, and it happens quite quickly. And they seem to persist longer than the microglial activation. And there's also evidence in humans that you get astrocyte activation in neuropathic pain. There was a study where they looked at individuals with HIV, neuropathic pain with HIV. Half of them had neuropathic pain, half of them did not. And they got post-mortem tissue and showed increased astrocyte activation in the dorsal horn. So we know that astrocytes are activated. We know that they can oscillate at roughly the same range that we're finding low frequency oscillation increases in our neuropathic pain patients. Now, one of the things that an increase in astrocyte activation would do is they get very large. One thing you can measure with an MRI scanner is water diffusion, or free water diffusion. So, and that's the whole basis of fibre tracking and all that kind of wham-bam stuff. So what it does is basically if you measure, if you take a voxel and you basically activate it in multiple directions, you can see how easily water moves in all of those directions. If the water molecule is in a ventricle, in CSF, it's going to move with Brownian motion, so it's going to move equally in all directions. If it's stuck between two fibres, it's only going to move parallel to the fibres, so you'll get a very strong movement direction, or easy movement direction along the fibre track, and that's how you can do fibre tracking. So what would happen with an increase in astrocyte activation is you would definitely have a decrease in the way in which water could move, because they get a lot bigger. So if astrocytes are activated in these individuals in the orofacial region, the orofacial or the spinal trigeminal nucleus, you should get a decrease in mean diffusivity. And we actually found that in orofacial pain patients. So you get a reduction in mean diffusivity, which is a measure of water movement, in the same area where you get an increase in these low frequency oscillations, and in the same area where you get increased astrocyte activation in humans with neuropathic pain. So it's all rather suspicious. Now, the other interesting thing, I was very thalamocentric for the last 5 years, and I thought if you lesion the thalamus, that would be it. And I sort of disregarded the dorsal horn to a large extent, or the spinal trigeminal nucleus. But you definitely get increased astrocyte activation there. You get, in humans, you get a decrease in mean diffusivity, which could occur because of an increase in astrocyte activation. You get an increase in infraslow oscillations. And it may be the case that they just pass them on to the thalamus, and the thalamus passes it on to the thalamic reticular nucleus, and that passes it on to the cortex. It may all be generated at the dorsal horn. I don't know. Or it may be a combination of the two. One thing we did do was to look at these low frequency oscillations between areas, and they are really tightly coupled. So they're not independent sort of signal changes. They seem to be tightly coupled in time, which would sort of imply that they're not independently generated. But in that study that I was showing you before, what they found, so they found an increase in low frequency oscillation in the thalamus after a CCI. They then did a spinal cord transection above the level of the CCI and found that these low frequency oscillations disappear. Now this could just be the fact that you've done a massive, lesion to the spinal cord, I don't know. But it sort of implies that these low frequency oscillations are being generated from the dorsal horn, which is sort of consistent with what we found. So we basically found altered anatomy up there, altered anatomy down there, The anatomy down at the spinal trigeminal nucleus might be indicative of astrocyte activation. We certainly have a change in the way in which the thalamus talks of the cortex and a decrease in the amount of GABA there. And we also have an increase in these sort of low frequency oscillations at sort of all levels of the chain, to be honest. At the spinal trigeminal nucleus, in the thalamus, and up at the primary somatosensory cortex. Now, I've been trying to work out how on earth thalamocortical dysrhythmia could actually give you chronic pain, and I have no idea. But there is really tight coupling between these low frequency oscillations and higher frequency activity. They're not independent. The reason why people never looked at these low frequency oscillations is because when they do EEG studies, they basically get rid of anything under 2 Hertz. But you can now do full band EEG where you measure. They basically thought it was noise. Well, a lot of it is, but they basically wouldn't measure it because it was noise. It was full of noise. So you can now do full band EEG. And when you do that, there's actually a really nice coupling between these sort of low frequency oscillations and activity or bursting time, or increases in high frequency activity. So they're not independent of each other. So I guess one of the things that we're thinking is that after you have a nerve injury, you get probably an increase in activity there that recruits astrocytes. You probably get an increase in these infraflow oscillations. They're passed up the chain to the thalamus. And in the thalamus, you get a sort of an alteration of this sort of thalamocortical circuit that involves somatosensory thalamus, thalamic reticular nucleus, and primary somatosensory cortex. And that basically ends up giving you a thalamocortical dysrhythmia. It may be that you have 2 different changes in rhythm. You have an infraslow rhythm that's coming up here, and the alterations you have here are basically altering higher frequency signals, and that interaction between those low and high frequency signals actually gives you a dysrhythmia. And that somehow maintains central pattern generator and chronic pain. That's where we're up to now. We're actually doing a lot of EEG work, trying to look at whether the infraslow oscillations are correlated to high frequency EEGs, and that's sort of where we're going down. The other thing that we're doing is looking at individuals with neuropathic pain, hoping to get them before they start any type of treatment and after they start some type of treatment. So to see if when you actually reduce their ongoing pain levels, do you get a reduction in thalamocortical dysrhythmia, do you get a reduction in these infraslow oscillations, and whether there's a link there between the constant perception of pain or the intensity of pain and their ongoing brain rhythms. Thanks.